So before we start to look at different bacteria that are pathogenic in humans, like the bacterium that causes tuberculosis or the bacterium that causes cholera, we just going to have a quick recap on the structure of a prokaryotic cell or a bacterial cell. So if we look at the diagram, we can actually see some of the main structures, such as the plasma membrane. Now we know the plasma membrane is quite simply a phospholipid bilayer, just like we've seen many times in different cell types. So it's a phospholipid bilayer made of phospholipids. Now we know each phospholipid is made of a polar head made of glycerol and phosphate, and two fatty acid tails that are hydrophobic, and there's two layers of phospholipids, and the fatty acid tails face each other, so it's the hydrophobic interior of the cell membrane, and the polar heads face out of the cell and also face in towards the cytoplasm. Now we know the cell wall in bacteria is made of peptidoglycan, and we know actually there's two main types of bacteria based on their cell wall structure. So the cell wall can either be of a gram-positive bacteria, and they will have a thick layer of peptidoglycan on the outside of the cell wall, or we've got gram-negative bacteria. So when you do a gram stain, the gram-negative bacteria will actually be stained pink because they retain the counter stain called safranin, whereas gram-positive bacteria will be stained purple because they retain the crystal violet dye. Now gram-negative bacteria have a very thin layer, so we can put thin layer of peptidoglycan, and that sits on the outside of the cell membrane. Then on the very outside of the cell wall, they have obviously the lipopolysaccharide rich, so lipopolysaccharide rich membrane on the very outside, and that affords them some protection from certain antibiotics such as penicillin. Now we also know some bacteria have a capsule or a slime layer on the outside, which is there really for protection too. Many bacteria have what are called pili, so these extensions that you can see here from the bacterial cell. Now pili are quite important in bacterial cells being able to recognize each other or bind to each other, so they're involved in cell-to-cell kind of recognition of the bacteria. But they're also important because sometimes when pili of different bacteria join together, they form little tubes down which plasmids can be transmitted from one bacterium to the next, and that is known as conjugation. So plasmids can actually be transmitted, the small circular pieces of DNA, between bacteria through pili that form these little tubes. Now we know the flagella, a bacterial cell might have one flagellum or many flagella, plural, and therefore the bacteria to be able to move through its extracellular fluid. Now we know inside the bacterium we've got cytoplasm, and that's where we get things like glycolysis taking place, the breakdown of glucose, the first stages of respiration. We get many enzymes therefore that catalyze these reactions. We get 70S ribosomes, so we know inside a prokaryotic cell 70S ribosomes, and that's where translation occurs. We get a large circular DNA molecule that is free in the cytoplasm. That's called the nucleoid. There's no nucleus, there's no membrane-bound organelles, because we know bacteria can be between 1 to 10 micrometers, okay, in length. So these are only small prokaryotic cells. These are about the same size as individual mitochondria or individual chloroplasts. They're around about similar size, between normally about 1 to 5 micrometers, sometimes a bit longer, 1 to 10 micrometers. Now we're going to start with tuberculosis. So the bacteria that cause tuberculosis are called Mycobacterium tuberculosis, and they are a bacillus type bacterium, and that means they are rod-shaped, as you can see in the picture here. This is a scanning electron micrograph, and we can see the outside of these bacillus tuberculosis causing bacteria in quite a lot of detail. There you go. So tuberculosis we know is caused by Mycobacterium tuberculosis, or M. tuberculosis, this bacillus bacterium, and we know it affects the lung tissue, actually the alveoli in the lungs, the little air sacs through which we get gas exchange between the air that we breathe in and the blood inside the capillaries that are on the outside of the alveoli. Now these bacteria can cause structures called tubercles. These little nodes of dead or damaged cells inside the alveoli, which is why the lungs get infected with people who have TB infection. So in this picture here we can see a gentleman with TB infection. Obviously he's lost an awful lot of weight. He's probably got blood in his sputum and very, very bad chest pains and coughing. Now if you take an x-ray you can actually see these tubercles inside the lung tissue, and here we've got some on the right side and here we've got some in the left lung of this patient, and it causes severe lung problems. Now these damaged cells, infected cells, can actually get into the lymphatic system of the patient, and it can also cause swelling of the lymph nodes. So we can start to think about linking some of the symptoms of TB infection with actually what happens to the bacteria once they get inside a person and infect that person. So three main symptoms then. One is obviously lung, chest pain, coughing up of phlegm or sputum, sometimes with blood in it. We know people with TB may get infected lymph nodes because infected white blood cells actually get into the lymphatic system. We get different types of immune reaction occurring in the lymphatic system, in particular inside the lymph nodes. So these tend to swell up. And we also get a fever. Now most pathogenic infections will give rise to a fever in the person because the pathogen will have evolved to replicate at a certain temperature. So increased respiration rate increases the temperature of the person to make it as inhospitable as possible for the pathogenic organism. So how does TB infection spread rapidly from person to person then? Well, we know the method or the mode of transmission is called aerosol transmission. Now the pathogenic bacterium M. tuberculosis is actually released from an infected person, we call this person number 1, in droplets of water when they get sneezed or coughed out. Now the bacteria in these droplets of water get breathed in by a second individual, and the bacteria travel down into the alveoli of the lungs. Now inside the alveoli, these bacteria get phagocytosed by white blood cells, in particular called macrophages or dendritic cells. So we get white blood cells that take in the bacteria to help break them down. But these bacteria have actually evolved a mechanism to be able to withstand phagocytosis within the white blood cell. So instead of actually being broken down by phagocytosis, they actually start to replicate inside the white blood cell. And that's what can cause these kind of nodules to form inside the alveoli of an infected person. Now these nodules, these infected white blood cells, they can also pass from the alveoli and the lung tissue into the lymphatic system of the person. Now we know aerosol transmission is going to be even greater when people are in crowded conditions. So we know historically TB has actually spread quite rapidly in highly crowded conditions, such as very, very densely populated cities. Now you don't have to know the life cycle of the bacteria that causes tuberculosis, the pathogenic bacterium, but it's interesting just to know a few little facts about this. So as we just said, these bacterium are breathed in through aerosol transmission, through little droplets of water. When they get inside the alveoli, they would normally be phagocytosed by certain types of white blood cell, for example macrophages, or there are other types called dendritic cells or monocytes. So these are phagocytic white blood cells. Now inside the phagosome or the phagocytic vesicle, inside the white blood cell, we would expect the bacterium to be hydrolyzed or broken down through enzymes like lipases, proteases, etc. However, these bacteria can actually survive inside the white blood cells and they start to replicate inside them. Now, more and more white blood cells get infected because they try to kind of get rid of the damaged white blood cells, and that can really result in this structure called a granuloma. Now, this is the collection of immune cells with, in the middle of the granuloma, infected white blood cells that are phagocytic cells. So you can see them here in the middle of the granuloma. So this granuloma can actually form inside the alveoli itself, which causes the kind of nodules to form, which reduces the effectiveness of gas exchange. So not only does it cause chest pain, it also causes breathing problems because you can't get enough oxygen from the air that you breathe in across the alveoli wall, those endothelial cells, squamous epithelium, into the blood capillaries. Now, not only do granulomas occur inside the alveoli, but they can also migrate and enter the lymphatic system. So inside the lymph nodes, so you can see here there are lymph nodes at regular sort of intervals or specific intervals throughout the lymphatic system. And this is where we get a lot of immune reactions to break down pathogenic organisms. So these granulomas can actually enter the lymphatic system and the lymph nodes, and they can cause swelling of these lymph nodes. Now, sometimes these granulomas can obviously burst open, and that releases more of these bacteria into the lymphatic system, so we get a spreading of the infection away from the lung tissue more extensively through the lymphatic system. So what would be the treatment for tuberculosis then? Well, we know it's caused by a bacterium, pathogenic bacterium. So the treatment's going to be antibiotics. Now, the regular treatment is a 6-month course of antibiotics in combination therapy. So you'll be given 2 antibiotics for the first 6 months. But for the first 2 months, you have an additional 2 antibiotics. Now, you don't need to know these names, so don't worry about learning these names. They're just there for reference. But in total, you have 4 antibiotics, all 4 for the first 2 months, and then 2 of them for the remaining 4 months to make it a 6-month treatment. Now, we do need to know why it's really important not only to take these antibiotics for the full 6-month period, but why we've actually got so many different types of antibiotic that we're taking here. So when we treat tuberculosis, we actually use 4 different types of antibiotic during the combination therapy, and not just one type. So why do you think this might be important? Well, each of the 4 different antibiotics actually targets a different process inside the bacterial cell. Now, we know bacteria are prokaryotic, so these different antibiotics can target, for example, translation, and that would occur at the 70S ribosomes in the cytoplasm of the bacterial cells. So it might block translation, protein synthesis. Some of the antibiotics might target transcription of DNA into messenger RNA. They might target formation of the peptidoglycan cell wall of the bacterium, or they might target DNA replication. So it makes the treatment more effective overall because the same bacteria are being targeted in 4 different manners. Now, we know DNA inside the bacteria replicates, and this is needed because obviously when bacterial cells divide by binary fission, the DNA gets distributed between both daughter cells. Now, when DNA does replicate at such a high rate, quite often mutations can arise within the new DNA strands. Now, we know mutations are spontaneous changes to the DNA occurring normally during DNA replication. Now, a gene is a sequence of codons whereby each codon codes for a specific amino acid in the polypeptide chain. So imagine one base was altered by a mutation, a random spontaneous mutation, and that gave rise to a different codon at that position. Well, that might mean an amino acid was different in the new protein strand or polypeptide chain, and it might give that protein a slightly different function. So it might mean that protein is resistant to the effect of the antibiotic. So this is how antibiotic resistance can emerge through spontaneous mutations in the bacterial DNA that can give rise to a slightly different version of the protein that might resist the effect of the antibiotic. Now, even if one of the genes that made the protein involved in whatever biochemical reaction we've got inside the cell, if that gene was mutated and the bacterium became resistant to the antibiotic that affected that new protein, it's very, very unlikely that these bacterial cells will have 4 separate mutations in 4 separate alleles that gives rise to 4 different proteins that this time are not affected by the antibiotic. So it's very unlikely that the bacteria will become resistant to all 4 types of antibiotic. So that's why this combination therapy is very, very effective. Now, in the text, it does say to complete the whole course of antibiotics, even though the patient may feel a bit better, only after 2 weeks. So 6 months is a long time to keep taking antibiotics. Well, this is needed because all the bacteria, the target bacteria that are pathogenic, must be killed. Now, if some bacteria survive and continue to divide, obviously, A, it's not going to get rid of the infecting pathogenic bacteria, but B, if they do survive and keep dividing for a continued length of time, it does increase the chance that those spontaneous mutations may arise. And if these spontaneous mutations occur in the alleles that give rise to a certain protein, if that protein has a slightly altered shape, but it can still perform its function, then the antibiotic might not be able to affect that particular bacterial protein, and therefore the bacteria might be resistant to the effect of the antibiotic. So we need to kill all the target bacteria, because if they're allowed to continue to divide, it increases the chances that some sort of spontaneous mutation might occur to make these bacteria antibiotic resistant. So preventative strategies for TB then. So preventative, we mean before we even pick up the pathogenic bacteria, what could we do to limit getting infected? Well, the BCG vaccine in the UK then, so these are given to babies, and the vaccine stimulates the immune response of the individual to raise antibodies not against the pathogenic bacterium, but against, in this case, an attenuated or weakened version of a very closely related bacterium from cattle called M. bovis. So M. bovis is a closely related species of bacteria that is quite similar to the pathogenic strain that causes tuberculosis. However, the closely related strain M. bovis does not cause tuberculosis in humans. It's non-pathogenic. So the immune system of the individual, the humoral response, will make antibodies against antigens on the surface of M. bovis. These antigens are actually quite similar to the antigens on the pathogenic bacterium. So these antibodies will actually recognize antigens on the infectious bacterium. If the individual does get infected with TB, they can raise an immediate immune reaction against the pathogenic bacterium, and therefore they can clear the bacterium before any disease can set in. So these people who get vaccinated against M. bovis become immune to infection by the pathogenic strain of TB bacterium. Now, a second type of bacterial pathogenic infection is called cholera. Now, cholera is actually caused by a bacterium that is present in sewage contaminated water supplies. So if people drink contaminated water, the bacterium goes through their digestive system and actually ends up in the small intestine or the ileum of that person. Now, most pathogenic bacteria secrete toxins, and the cholera bacteria is exactly the same. And this toxin actually results in a very, very watery diarrhea. So people with cholera tend to actually die of dehydration because of this very, very watery diarrhea. And obviously it does result in very, very rapid weight loss and very, very rapid dehydration, which is what kills the infected person. So if we have a look at this slide, we can actually just start to fill in the blanks here if we read through the paragraph. So it says cholera is caused by a comma-shaped gram-negative bacterium. That means if you stained the bacterium with a gram stain, at the very end of the process they would appear a pink colour because they retain the counterstain safranin. And the bacterium is called Vibrio cholerae. Now, it can only reproduce when it's inside the human small intestine, i.e. the ileum. So we can put here ileum. And that's where most digestion and therefore absorption occurs from the human diet. Now, cholera is, keyword, in certain areas of the world, such as parts of Africa and Southeast Asia. Now, that keyword is called endemic. And that means that this cholera is actually present in low levels in populations in these areas. And it's been there for an awful long time. Now, people do become infected through contaminated water and also sometimes contaminated food. So imagine vegetables have been washed in contaminated sewage water. Well, obviously the bacterium is going to get on the vegetables and then they get consumed into the ileum, small intestine. Now, any people who do become infected with TB, it can be, or they're called disease carriers and disease reservoirs for this particular pathogenic bacteria. And they can contaminate other water supplies, which results in a spreading of the disease. Now, just before we talk about the mechanism of how the pathogenic bacteria actually causes cholera, just going to do a brief recap of some of the material from year one. So how do we define water potential? Because we're about to talk about how watery diarrhea is formed. And it involves water potential ideas. Now, water potential is quite simply the potential or the tendency for water to enter or leave a cell across a partially permeable membrane by osmosis. And that's going to be down a water potential gradient from a less negative to a more negative water potential. So water potential is the potential energy or the tendency for water to enter or leave a cell by osmosis across a partially permeable membrane down a water potential gradient. Now, there is an equation for water potential, which is the water potential of the cell is equal to the solute potential plus the pressure potential of the cell. However, this is only applicable really to plant cells because plant cells have a cellulose cell wall, which we know creates a positive pressure potential when the cytoplasm expands and pushes against the inelastic cell wall. Well, we know animal cells do not have a cell wall. So we can put that here, have no cell walls. So this equation that we use for water potential is not applicable to animal cells. So this is the mechanism of how the toxin produced from the cholera bacteria results in a watery diarrhea. Now, these bacteria, once they get into the ileum, they can start to replicate by binary fission, and they do secrete this toxin. So these black circles here, we could put a T inside maybe, these are actually the toxin molecules. Now what they do is they bind to a glycoprotein receptor on the surface of the epithelial cells that line the length of the ileum. So lining the villi down the length of the ileum is a layer 1 cell thick of epithelial cells. Now the toxin binds to the glycoprotein receptor on the surface of the epithelial cells that line the ileum, and that causes an intracellular signal to be sent to a chloride ion channel protein in the membrane. So this is like a signal sent inside the cytoplasm that switches on the chloride ion channel protein. Now, because this chloride ion channel protein, to put here, chloride ion channel protein is switched on, it means that chloride ions will actually move out of the epithelial cells lining the small intestine into the lumen. So this is the space on the right hand side within the small intestine. Now the chloride ion channel protein is actually called the CFTR channel protein, and that really means cystic fibrosis transmembrane regulator. Now you don't have to remember that name, but as you can imagine, this is actually a protein that's also involved in cystic fibrosis. And in CF, or cystic fibrosis, there's actually a mutation in the allele or gene that codes for the protein. So there's a shortened non-functional chloride ion channel in cystic fibrosis patients. So they actually have the opposite effect. They can't get chloride ions out of the epithelial cells in their trachea, so they actually end up with a very, very thick, sticky mucus. But here, the chloride ion channel, the CFTR protein, is actually always switched on. So it's always going to be open because of the binding of the toxin to the receptor. So we put here receptor at the top. Okay. So these chloride ions move out of the epithelial cell. They concentrate in the lumen of the small intestine. And we know when you've got more solutes, or ions in this case, that become more concentrated, it drives the water potential to a more negative value. So the fluid inside the lumen of the small intestine becomes hypertonic, i.e. more negative in water potential. And obviously the units would be kilopascals. Now, there's also sodium ion channels within the membrane of the epithelial cells. Now, when they are open, now the sodium ions move out by facilitate diffusion towards the negative charge of the chloride ions in the lumen of the small intestine. And that further contributes to the negative water potential value. So it drops even lower, more and more negative water potential in the lumen of the ileum. Now there are another type of channel protein called aquaporins. Now when we study the kidney, we know aquaporins are channel proteins that allow the movement of water into or out of a cell. Now water can't really just diffuse simply out of the cell, because we know water is a polar molecule, and we know the hydrophobic fatty acid tail to the membrane would prevent the simple diffusion of water out of the cell. So water, because it's polar, has to go through a channel protein called an aquaporin. And we know water always goes from a less negative to a more negative water potential. To a more negative water potential value down a water potential gradient. So water moves down a water potential gradient from inside the epithelial cells into the lumen of the small intestine, and we know that is by osmosis, and that's why you get the watery diarrhea that is so characteristic of cholera. Now the treatment for cholera is actually not just to give water to rehydrate them, but rehydration solution, and that solution should actually contain both sodium ions, i.e. salt, plus glucose, plus obviously water in the solution. So you want to treat someone with cholera, give them a salt and glucose solution, because the sodium ions and the glucose actively get co-transported into the epithelial cell, as we've done in the past in digestion. And that makes the water potential of the inside cytoplasm of the epithelial cell slightly more negative. So it's going to prevent too much water escaping from the epithelial cell. So the treatment is rehydration solution with sodium, glucose, and obviously water.